Hello, welcome. Today, we're going to talk about a microscopic parasite that shapes our planet's microbial world. Many of you have heard of this warrior, but some may not know just how useful they are. Others might be scared of them, since some, their closest relatives, have a reputation for causing harm to our health, as well as the health of animals and plants we care about. This tiny but powerful entity is a virus called a bacteriophage. Bacteriophages, or phages, the viruses that infect bacteria, are the most abundant biological entities on Earth, with an estimated 10 nonillion on the planet. They outnumber the stars in the observable universe by 1 billion to 1. These microscopic entities are so plentiful that they are estimated to outnumber every other living thing on the planet combined. The estimated total biomass of all bacteriophages on Earth ranges from 70 to 100 gigatons of carbon. In fact, this exceeds the estimated biomass of all humans, whales, and elephants combined. Their name, bacteriophage, literally means bacteria eater, derived from bacteria and the Greek word phagein, meaning to devour. They are nature's viral hunters, targeting bacteria with precision, unmatched by any other microorganism. Structurally, bacteriophages are relatively simple, especially when compared to the complexity of cells or other life forms. They typically consist of a protein capsid, a protective shell that encases their genetic material, which can be either DNA or RNA. Many phages have a distinctive tail structure, like a microscopic syringe, which they use to inject their genetic material into a host bacterium. Other types, called tailless phages, rely on different mechanisms to get their genetic code into their target. While the majority of bacteriophages have a tail, it's not always necessary. The diversity in their structure is astonishing. Some phages look like alien landing pods with spindly legs, while others resemble geometric crystals. This structural variety is just one example of the incredible evolutionary diversity phages display. The infection process of a bacteriophage is nothing short of fascinating. It begins with a highly specific recognition stage. A phage searches for its preferred host, identifying specific receptors on the bacterial cell's surface. These receptors can be unique proteins, lipopolysaccharides, or even components of the bacterial flagella. This specificity is so precise that many phages will infect only a single bacterial species, or even a single strain within that species. Once attached, the phage injects its genetic material into the bacterium. At this point, the virus has two choices: the lytic or the lysogenic cycle. In the lytic cycle, the phage takes immediate control of the bacterial cell's machinery, using it to create copies of its genetic material and produce new viral proteins. As more and more viral particles are assembled, the bacterial cell fills to the breaking point and eventually bursts, releasing a flood of new phages to continue the infection. In contrast, the lysogenic cycle is a quieter affair. Instead of destroying its host, the phage's genetic material integrates into the bacterial genome, lying dormant. This integrated DNA, known as a prophage, can be reawakened by environmental triggers, like UV light or certain chemicals. When this happens, the virus switches to the lytic phase, creating a burst of new phages and killing the host cell. However, not all bacteriophages kill their hosts during their reproductive cycle. Some have evolved ingenious strategies to exit their bacterial hosts without causing immediate destruction. Certain filamentous phages use a mechanism called extrusion. These phages replicate within the host cell without causing lysis. Instead, they utilize the host's pili, tiny hair-like appendages that many bacteria use for attachment or DNA transfer, to extrude themselves out of the cell. In this way, the bacterial host remains alive and continues to produce more phages over time. This is akin to a parasite that can live within its host indefinitely, drawing resources without causing death. Another method involves a process similar to how some animal viruses operate, which is through budding. In this case, the phage particles slowly bud off from the cell membrane without causing immediate damage. This method allows the bacterium to survive and continue functioning while still serving as a factory for new viruses. Bacteriophages are not just microscopic assassins, they are key players in Earth's ecosystems. They play an essential role in regulating bacterial populations, keeping microbial communities in balance, and contributing to the cycling of nutrients. Phages are constantly at war with bacteria, driving bacterial evolution. In response to phage attacks, bacteria have developed mechanisms like the CRISPR-Cas system, a form of genetic memory that allows them to recognize and defend against past phage invaders. This system has since been harnessed by scientists as a powerful tool for genetic engineering. In recent years, bacteriophages have been attracting renewed interest for their potential in medicine, especially in combating antibiotic-resistant bacteria. This approach, known as phage therapy, involves using specific phages to target and destroy pathogenic bacteria without harming beneficial ones. Unlike broad-spectrum antibiotics, phage therapy offers a precision strike, reducing the risk of collateral damage to the body's microbiome. Research in this field is expanding rapidly, with clinical trials investigating phage therapy for conditions like chronic wounds, respiratory infections, and gastrointestinal diseases. Some countries like Georgia and Russia have used phage therapy for decades, while Western medicine is only beginning to catch up. Bacteriophages have a variety of uses beyond therapeutic applications. One of the most important is phage display, a molecular biology technique where phages are engineered to express proteins on their surfaces, allowing researchers to study protein interactions. Phage display has applications in drug discovery, vaccine development, and understanding protein functions. In the food industry, phages are used to enhance food safety and preservation by targeting specific pathogens, such as Listeria and Salmonella, reducing the risk of foodborne illnesses without affecting the taste or quality of the food. Phages are also employed in agriculture as natural biocontrol agents to target harmful bacteria that infect crops or livestock, offering an eco-friendly alternative to chemical pesticides. In wastewater treatment, phages help reduce bacterial contamination in sewage and industrial waste by specifically targeting harmful bacteria, thereby improving water quality. Additionally, phages are utilized as diagnostic tools for rapidly detecting bacterial infections due to their high specificity for certain bacterial strains. In biotechnology, phages are valuable in gene cloning, sequencing, and genetic engineering, as they can efficiently deliver foreign DNA into bacterial cells for research and industrial applications. Phages also play a role in environmental monitoring, helping to detect bacterial contamination in water sources, food production facilities, and hospitals. In livestock management, phage therapy is used to control bacterial infections in animals, reducing the need for antibiotics and helping to prevent the spread of antibiotic-resistant bacteria. These diverse applications illustrate the broad potential of bacteriophages in a wide range of fields. Bacteriophages and bacteria have been co-evolving for billions of years. The oldest known traces of viruses date back to nearly 3.5 billion years ago, suggesting that bacteriophages may have existed since the dawn of life on Earth. This ancient evolutionary relationship is a testament to the ongoing arms race between host and parasite. Bacteriophages are not only destroyers, they are also genetic messengers. They can facilitate horizontal gene transfer, moving genes from one bacterium to another. This is a key factor in bacterial evolution, as phages sometimes inadvertently transfer genes that provide bacteria with new capabilities, such as antibiotic resistance or virulence factors. Bacteriophages can be found in a wide range of habitats wherever bacteria are present. They are abundant in soil, where they help regulate bacterial populations. Phages also thrive in water, including both freshwater and marine environments, playing a crucial role in controlling bacteria in these ecosystems. In the human and animal bodies, phages naturally exist in places like the gastrointestinal tract, skin, and respiratory system, interacting with and regulating bacteria. They are also commonly found in wastewater and sewage, where they help break down harmful bacteria. Phages can survive in extreme environments such as hot springs, ice-covered lakes, and deep-sea hydrothermal vents, where they infect bacteria adapted to harsh conditions. It is estimated that in just 1 milliliter of seawater, there are up to 1 billion bacteriophages. In fact, the oceans are the largest reservoirs of phages, where they play a pivotal role in controlling bacterial populations shaping marine ecosystems and influencing global nutrient cycles. Bacteriophages represent a vast and largely unexplored realm of biological diversity. Scientists believe there are likely tens of millions of unique phage types, each adapted to a specific bacterial host. This genetic diversity makes phages a potential goldmine for discovering new enzymes, molecular tools, and biotechnological applications. Some phages possess unusual properties, like encoding their own unique enzymes to degrade bacterial cell walls or hijacking the bacterial machinery in novel ways that are still poorly understood. As our understanding of bacteriophages deepens, their potential applications in medicine, agriculture, and industry continue to grow. Phage-based diagnostics are being developed to detect bacterial infections rapidly, while phage enzymes are being explored as biocontrol agents in agriculture to target crop diseases. The world of phages is vast, mysterious, and filled with untapped potential. As we continue to unlock their secrets, bacteriophages could become one of our most powerful allies in medicine and biotechnology, revolutionizing the way we manage bacterial infections and interact with the microscopic world around us.